Assalamu alaikum, ladies and gentlemen. Today I'm going to present uh, my talk on a topic. Actually, this is not a talk, this is actually a workshop. And this is the work that I have completed over the last 5 to 6 years. We have developed this system here in Punjab University where we actually work on the in silico genome-wide analysis of different gene families in plants. So, and as this workshop is related to genome-wide analysis, especially against bacteria, so I will tell you at the later of this slide how we can use this technology to understand the functions in plants in and also in bacteria. So, our, first of all, I will tell you about what is bioinformatics. Bioinformatics is a new branch of science that integrate plants, biology, microbiology, zoology with computer science, and also then in bioinformatics we actually study data science in bio. So I normally call that this is known as the biodata science because whatever the data that is generated in biological science, we actually analyze this data and ask the computer to make some algorithms that you can easily understand the pattern in biological data and how we can get information from that data. So as I already told you that in bioinformatics, we normally did many things like omics studies, phylogenetic analysis, understand the sequence homology, and then understand the transcriptomic, metabolomics, and proteomics of different plant cell under different response. So, and from all these studies, we actually analyze this data, and that will help us to understand the information from that data, and we are able to have some surprising conclusions from that data. So, what are the table of content of this today's talk? First of all, I already told you about the introduction, what is genome-wide analysis, and then we will use the different techniques that are normally studied during genome-wide analysis using computational biology, and then we will go some functional analysis and regulatory studies, and ultimately we will go over some evolutionary studies that we can perform using in silico genome-wide analysis. So, what is transcriptome analysis? It is very important technique that is used by the researcher to understand the expression of the genes and the function of the genes and help us to understand the differentially expressed gene. From these data, we can easily interpret that what this gene can do during that specific stress or during that specific organ. So, and then we are also tell you about the applications and implication of this genome-wide analysis using in silico approach for the understanding the various field and future direction because of the add of AI and machine learning tool. We are also moving towards that. So, what is a genome-wide study? During the genome-wide study, we actually scan all the genome of plants, animals, bacteria, and use that genome to identify the different genes and the different variation that are present in the gene. And this is very important at step for understanding the physiology and molecular biology of the any organism. So what we actually did during genome-wide studies is the gene discoveries, gene functions, and gene genetic diversity and evolution of that gene. So molecular biology, we understand the gene regulation, gene network, gene functions, and their interaction with different other genes and other proteins and other metabolites. We can study that. And then functional genomics. This is very important to understand the function of the gene. This in silico approach help us to at least predict the functions of the gene by analyzing the different pattern in DNA sequence, protein sequence, and then we also study some regulatory mechanism like expressions of the gene, and then there are structural analysis that gene structures, how they are different members in the same family are different and how they are interact with each other. And next is no, the question is that how you can design a research plan or projects or paper so that you can use genome-wide in silico approach to understand the gene and in the specific crop, animals, insects, or bacteria. First of all, you need to understand the you must have know what are the specific objective that you have in your mind to understand the function of the gene. So normally what we actually do, first of all, we identify the gene that has not been characterized in that specific organisms. For that purpose, we actually survey the different research papers present in Google Scholar and online Google survey. After the end of that survey, we are able to find that this gene family has not been characterized in this crop or in this animal or bacteria. So our next purpose is whether is there any specific objective that are we are following to understand the function of the gene, like is the crop has specific problem like drought stress, heat stress, or is there any pathogen that actually targeting this crop and reducing the yield of the plant? What are the microbial attack or the microbial infestation that reduces the yield of the plants or animal? So for that purpose, if after all analyzing all these aspects, we actually then design a specific objective of the research plan and then target the stress conditions. So for that purpose, then we after analyzing that, we will work on this gene and on this crop, and we will solve this problem in this crop by identifying the gene function and their gene family and their gene regulation, so that that whole analysis tell us that how this gene behave during that specific stress in that specific organisms. So we for as we know that this is the gene of the name, and then we actually use different genome-wide analysis software to understand the function of that gene. So this is the some of the work that we have already published and we have already done to understand the genome-wide in silico expression as a phenylalanine amine lysase gene family. This gene family is a chitinase gene family. What is a chitinase? We all know that chitinase is a gene that actually degrades the chitin. Normally, this chitin gene is present in different fungus fungi. So our purpose is to understand why this gene is present in plant, what is the function of that gene, can this gene be used to control the fungal pathogen in plants? Because these chitinase gene play a vital role in the enzymatic degradation of pathogenic chitin through hydrolysis. So this gene family assumed to be a significant defense related against pathogen, and also this gene is reported to be involved in abiotic stress response. So for that purpose, we actually gene identify the PAL gene and then identify their defense mechanism, and then apply. We will apply this gene to understand the functions in different plant species. So PAL gene is I already told you that it's very important, that is involved. That is a major part of a stress-related mechanism in plant. So what now how we can identify the gene? First of all, we need to know what are the domains that are present in the protein of this gene. So PAL gene that encode enzymatic phenylalanine ammonia lysase. So PAL catalyze the deamination of L-phenylalanine to trans-cinnamic acid, which is the first step in phenyl uh, 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 polypeptide pathway. So this pathway leads the production of various secondary metabolites. So that secondary metabolites help the plant to understand the function of the gene. And okay, so similarly, this genome-wide analysis of PAL with literature, survey the literature and understand the function of that gene by so key techniques, explanation of the phylogenetic analysis and its comparison with the evolutionary relationship, and understand how this gene is present. If we are working in some specific problem like in sorghum, if we find that there are 10 genes of PAL in sorghum, why there are only 10 genes in there? What are whether this gene is present in other crop as well? If some other crop has more than 10 genes, like 20, 25, or 30, and will also then understand whether there are some other crops that can have Paul gene less than 10. So our understanding, first of all, why these genes are not so much in that specific plant, not in so much number. It means that this gene is not as much evolved in that gene as compared to other genes. And then we will also identify different regulatory network of this gene in that specific crop. Maybe this gene has developed in a different way, and they do some different jobs in plants, that specific plant, than the other plants. So we actually analyze all these things. We use different softwares, MCBI, Photoshop tool, Vega, TV tools, but I cannot explain this in just 1 hour. For that purpose, we need a lot of time and a series of lectures so that we can understand how these different online website tools you can be used to understand the gene family evolution and their functional divergence. So first of all, we actually made the phylogenetic tree. Most of the people, they know what is a phylogenetic tree. Phylogenetic tree help us to understand the relationship between the ancestors and their descendants. So based on that analysis, we actually group the genes into different families and group based on their evolutionary history. So, and what are the elements of phylogenetic tree? There are roots, branches, nodes, clades, and bootstrap values. We are normally value shows that the branch of the tree that indicates the level of sports for a particular clade. So, high bootstrap value indicates strong sport. Similarly, we also did some physiochemical properties of all genes by extracting this from online databases, like we did the gene ID, sequence length, PI value of the genes, and then also retrieve information like the genetic from where gene start, from where gene starts, what are the location of the chromosomes. So this is the data we actually extract from different websites, and how we extract that data, I will tell you detail in some other videos. So what are the key protein properties? The total mass of the molecule, that is known as the molecular weight. pH, where the molecule has no net charge, is very important for protein purification and also the functioning of the protein. And instability index predict in vitro based on amino acid composition and grand average of hydrophobicity, measure of hydrophobicity and hydrophilicity. The positive gravity means more hydrophobic and negative gravity means more hydrophilic. So, and then we also did some another analysis like from where this gene is present in the cell, in the subcellular localization, what happened to the gene when they, after the ribosomes, where they will go. So we can do this analysis by using online Evolve P-sort and Target P website. And this is a database that is specifically designed for subcellular localization, which help us to understand the protein function and their interaction within the cell. So this is this we have done this whole hologram vulgare. So that is actually genes location, subcellular localization, and from that this gene is present mostly in this chloroplast, and some other genes are have their presence in some other part of the plant of the yes of the cell. So similarly, we also do some intron exon. Every gene they evolve differently. They might lose some intron. They might add some exons. So that will help. That may change their functions, or they may help the plant to adopt under different climatic condition. Because today is a day of climate change. Everyone in the world they work on the climate change, so they write a project based on climate change. So in this way, we can easily understand why this gene they evolved under different climatic condition, why this intron exons pattern are actually helping the plant to control different things. So and we actually do this functional analysis using intron exon structures, and similarly we do some other analysis as well.